minutes. Lakas ng sound.
Okay. Hello and good evening, everyone. Okay. Okay. So hello and good evening, everyone. Welcome to the third Philippine Herbal Medicine Summit. As general reminders for everyone, to all participants, please use your first name and last name as your username for easy identification. Please ensure that your microphone is on mute and your web camera is turned off throughout the duration of the summit. Questions will be entertained at the end of each plenary session, and please use the raise hand function at the sidebar if you have questions, or simply type your question in the question and answer chat box. Please monitor the chat box for any announcements. We will now proceed to the invocation followed by the national anthem.
Praise God from whom all blessings flow. Praise Him, all creatures here below. Praise Him above, ye heavenly hosts. Praise Father, Son, and Holy Ghost.
Mga kababayan, ang pambansang awit ng Pilipinas. Kayang magiliw, helas ng sinanganan, alam ng puso, sa titik mo'y buhay.
Good evening. We are very so fortunate today to have our dear Executive Director of the National Institutes of Health, UP Manila, to officially open the third Philippine Herbal Medicine Summit. To give us her welcome message is Dr. Eva Cuchonco de la Paz, one of our heroes in genetics and multi-awarded Executive Director of the National Institutes of Health.
FDA Director General Dr. Eric Domingo, Professor Chun Tao Che from the WHO Collaborating Center for Herbal Medicine, College of Pharmacy, University of Illinois, Professor Fabian Dairit from the Ateneo de Manila University, Vice Chancellor of UP Visayas Dr. Philip Ian Padilla, our other distinguished speakers, and of course, special greetings to UP Professor Emeritus Dr. Nelia Cortez Maramba and NIH Institute of Herbal Medicine Director Dr. Cecil Maramba Lazarte, our IHM research faculty, staff, guests, friends, and colleagues. Welcome to the third Philippine Herbal Medicine Summit. The theme of this summit, ensuring quality of herbal medicine through research, regulation, and cooperation, is very relevant as it brings together all herbal medicine stakeholders, including researchers, regulators, manufacturers, distributors, funding institutions, and consumers to discuss herbal medicine's current status during the new normal and beyond. Our speakers from both the government and private sectors will share their plans on herbal medicine research, regulation, and cooperation. Our host, the NIH Institute of Herbal Medicine, has prepared stimulating activities embedded in this conference, such as research contest, an interesting TikTok contest, and a talk on the Philippine Herbal Pharmacopoeia set to be released in 2022. We have exciting times ahead as herbal medicines have been in great demand. The IHM and its partners are doing their very best to make herbal medicine accepted as mainstream medicine by providing rigorous scientific and clinical validation to prove their safety and effectiveness. In the very near future, the opening of the new NIH building will greatly contribute to herbal medicine development as it will have an animal research center, the NIH Clinical Research Center, and a formulation unit at the Institute of Herbal Medicine. The Institute's mission to have more effective but inexpensive quality medicine with better cultural acceptability available to every Filipino is truly within reach. Again, welcome, and may we have a productive conference ahead. Magandang gabi po sa inyong lahat.
Thank you so much, Dr. Eva, and taking so much time out for on your very, very busy day and still to be with us here this evening. So now it is my pleasure to introduce to you our keynote speaker for the 13th Philippine Herbal Medicine Summit. He is currently the Director General of the Philippine Food and Drug Administration and Undersecretary of the Department of Health. He was a graduate of the UP College Medicine Class 1992. Kami yun. Walang katulad, Class 1992. He finished ophthalmology residency at the Philippine General Hospital and a fellowship in ophthalmic pathology and oncology in the Universidad de Valladolid in Spain. So let us all welcome Dr. Rolando Enrique Domingo.
Magandang gabi po sa inyong lahat. Good evening, especially to my friends and co-workers, the Executive Director of the UP National Institutes of Health, Dr. Eva Cuchonco de la Paz, my beloved classmate, Dr. Cecil Maramba Lazarte, to the plenary speakers, guests, and attendees. thank you for having me here tonight as we celebrate the third Philippine Herbal Summit. Over the years, herbal medicines have been an important part of our lives and culture as an alternative way of addressing health concerns. With a desire for natural alternatives, people usually refer to medicinal plants for their healthcare choices. Research is continuously being conducted to strengthen the scientific evidence on the therapeutic value of herbal products. The role of the National Institutes of Health, UP, and other universities like Ateneo is invaluable. With desirable patient outcomes, herbal medicines have become important therapeutic options, and medicinal plants are now important sources for pharmaceutical manufacturing. As a regulatory agency mandated by Republic Act 9711, it is the duty of the Food and Drug Administration to ensure the safety, efficacy, and quality of health products for public consumption. Herbal medicines are part of traditional medicines and fall under the category of pharmaceutical products. It is our mandate to ensure that herbal medicines are regulated through licensing, registration, and post-market surveillance. Compliance to good manufacturing practice remains to be one of the most important requirements to ensure safety, efficacy, and quality. Advertisement and promotion must be based on approved product registration, indication, and proper use. The herbal medicine industry must work with us to ensure that identification, evaluation, preparation, and cultivation of medicinal plants are based on science and clinical evidence. All are advised to ensure quality control of herbal medicine on a batch-to-batch basis using modern techniques and applying suitable standards and GMP. As part of our continuous growth, the FDA participates in ASEAN meetings that discuss technical guidelines and regulatory requirements for member states to implement. The various forms of traditional medicinal products have evolved against widely different ethnological, cultural, climatic, geographical, and even philosophical backgrounds. The evaluation of these products and ensuring their safety and efficacy through licensing and registration remains to be a challenge, but we assure you that we are doing our best to realize the goal of harmonizing with our ASEAN neighbors. The Philippine Institute for Traditional and Alternative Healthcare, or PITAC, works to facilitate the integration of traditional medicine into the national healthcare system, promote the rational use of traditional medicine through the development of technical guidelines and international standards in the field of herbal medicine, and act as a clearinghouse for the dissemination of information on various forms of traditional medicine. The FDA, on the other hand, will work to ensure systematic inventory and assessment of the medicinal plants used in herbal medicinal products. We will continue to study and try to introduce measures for the regulation and control of medicinal plant products and for the establishment of suitable safety, efficacy, and quality standards. I look forward to hearing more from you after this third Philippine Herbal Summit. Let us work together to protect public health safety and welfare. As you are well aware, the FDA is full steam in implementing Republic Act number 11032, or the Ease of Doing Business and Efficient Government Service Delivery Act of 2018. The FDA is undergoing continuous streamlining of its licensing and registration requirements and processes. To close, I would like to emphasize that the FDA is your partner in promoting the right to health of all Filipinos. We are firm with our mandate in ensuring that health products are safe, of good quality, and of good efficacy. We are streamlining our requirements and re-engineering our processes and systems to give better services. Let us work together to give our fellow Filipinos the best health products that they deserve. Thank you, and I hope you have a fruitful event ahead. Maraming salamat po.
Thank you so much, partner Eric.
Hi,
So,
Cecil. Actually, I'm here.
hi, hi. Hi! Nice to... Buti, hinahanap nga kita dito sa participant. Thank you so
Yeah.
much for being with us.
Yeah.
Nakahabol ka.
Oo, gusto ko lang dagdag. Si Dr. Cecil and I were also working on the Philippine Herbal Pharmacopoeia. And, there's so many things to do with our herbal products. And you all have the support of the FDA. And I really wish to be working a lot closer, lalo na pag natapos na yung pandemic.
Yes, kaya nga sabi ko na, partner Eric. So thank you so much, Eric. We appreciate you're here. And we would like to present to you this certificate of appreciation for your invaluable contribution as our keynote speaker for the third Philippine Herbal Medicine Summit with the theme ensuring quality of herbal medicines through research, regulation, and cooperation today on January 25 during our Zoom webinar. Thank you so much. So we will have a picture taking with
Thank you.
Dr. Eric. Teka, mag-picture muna tayo with Ma'am Eva and our other speakers today. SL, magpipicture tayo.
So may we request everyone to please turn on their cameras for a short photo op with our speakers. Thank you.
Ma'am Eva, ayan. Okay, dapat ba gallery? Ayun. Okay. Okay, smile everyone. 1, 2, 3. Got it, SL? Okay. Thank you so much, Ma'am Eva. Thank you po, Sir Eric. Okay, Paolo?
Okay, hello and good evening. Again, I'm Dr. Paulo Cerrado, and I'll be moderating the proceedings for today. We will now proceed to the plenary symposium, which is the initiatives to improving standards for herbal medicines. We'll be joined by Jocelyn Palakpak to discuss the Philippine Herbal Pharmacopoeia. Professor Palakpak is a professor at the Department of Industrial Pharmacy, College of Pharmacy at the UP Manila. She is also the Assistant Director of the Institute of Herbal Medicine, National Institutes of Health, and the project leader of dosage forms of Philippine medicinal plant constituents, which is the formulation group of the National Integrated Research Program on Medicinal Plants, or NIRPROM. She obtained her BS Industrial Pharmacy and Masters of Pharmacy at the University of the Philippines, Manila, and she is also the former Dean of the College of Pharmacy of the University of the Philippines, Manila. Our next for our plenary is Dr. Chun Tao Che.
The first time I've encountered a pharmacopoeia was during my second year in college. I had no idea how meticulous a pharmacopoeia was prepared. All I know at the time is I've got to finish the pharmacognosy worksheets or else Dr. Cantoria won't let up during recitations. Good evening. My name is Jocelyn S. Bautista Palakpak, and I will narrate to you how we at the Institute of Herbal Medicine are preparing the Philippines' first herbal pharmacopoeia. Here's the flow of my story. First, I'll give you a brief background on the pharmacopoeia. Then I'll be discussing the Philippine Herbal Pharmacopoeia project. Finally, I'll let you in on our future activities and plans. So what is a pharmacopoeia? A pharmacopoeia is a compendium describing medicinal products issued by an officially recognized authority and serving as a standard. It's a legally binding collection of standards and quality specifications for a medicine in a country of origin. So what are pharmacopoeial standards? These are publicly available compliance specification. They provide the means for an independent check of the quality of a product at any time during its shelf life. Because the foremost objective of a pharmacopoeia is to protect public health through standards to ensure the quality of medicines. As of 2015, the WHO has recorded 49 pharmacopoeias all over the world. They include the International Pharmacopoeia, which is published by the WHO, the United States Pharmacopoeia, the British Pharmacopoeia, the Japan Pharmacopoeia, and to give an example, there are also herbal pharmacopoeias such as a Thai Herbal Pharmacopoeia, which is annually published, the African Herbal Pharmacopoeia, and the American Herbal Pharmacopoeia. And of course, hindi naman tayo papahuli. We also have the Philippine Pharmacopoeia 1. Okay, so let's talk about the Philippine Pharmacopoeia 1. The project was initiated by JICA in 1999 and was finished in 2004. And with Executive Order 302, it was adopted as the official pharmacopoeia, the official book of standards and specifications. It consisted of two parts. Part 1 contains 17 monographs about synthetic medicines with 30 medicinal preparations, while Part 2 consisted of 29 monographs on medicinal plants with 22 powdered plant materials. Most of the basis for Part 2 is folkloric use. So that was in 2004. Now it's 2021. So how old is the pharmacopoeia? Next year, magdidibo na siya. Okay? So you expect a lot of developments in healthcare, in science, and technology. Plus the fact that herbal medicine has been gaining more popularity throughout the years. And this is evidence, I think, by the pictures that were chosen for this slide. I think this is the first, what you see on the slide is the first, let me backtrack. The first page of the Philippine Pharmacopoeia, the original cover is green, but I guess the cover page is already not good to look at. And then we have 3 pages here of the composition of the pharmacopoeial organization, from the executive board to the steering committee and members of the technical working groups at the time. Okay. Now we go to the Philippine Herbal Pharmacopoeia project. Okay, this is a project under the Institute of Herbal Medicine, or IHM. We started talking about the PHP, the Philippine Herbal Pharmacopoeia, when we did our strategic planning in 2016 and 2018. And in 2019, we presented a proposal for the project, and happily, PITAC, or the Philippine Institute of Traditional and Alternative Healthcare, said yes and provided the funding. Okay. The general objective of the project is to develop and produce an updated pharmacopoeia of Philippine herbal medicines and medicinal plants containing 45 monographs of medicinal plants and herbal medicine. Its specific objectives include the creation of the pharmacopoeia organization, to prioritize the medicinal plants and herbal medicines which will be included in the PHP, to set the format, official standards, references, and tests to determine the identity, purity, and quality of drug products and crude plants, and to collect and process the medicinal plants and perform the appropriate tests specified by the committees. The target beneficiaries, first and foremost, are the Filipino consumers, the researchers and practitioners. Also, the Philippine Food and Drug Administration and the Philippine pharmaceutical and herbal industries. This slide shows you the activities of the project. So the ultimate goal is to have the Philippine Herbal Pharmacopoeia declared and adopted as the official book of standards and references. Similar to what happened with the Philippine Pharmacopoeia 1. Okay. So to get things going, the Pharmacopoeia organization has the following committees: the Executive Committee, the Steering Committee, the Technical Working Group 1, and Technical Working Group 2. The Executive Committee has the following responsibilities: provide overall policy direction, establish funding sources, approve recommendations of the Steering Committee, review and approve the recommendations of the technical working group, and ensure the continued development and revisions of the Philippine Herbal Pharmacopoeia. So we have the following personalities comprising the Executive Committee: Dr. Eric Domingo, sitting as the chairman of the Executive Committee. He's also currently the Director General of FDA. Dr. Annabel P. de Guzman, co-chairing. She is the Director General of PITAC. Attorney Sherade B. Mercado Grande, Attorney Carmencita Castro Santos, and Dr. Cecilia C. Maramba Lasarte. Then we have the Steering Committee. Okay. They supervise and support the work of the technical working groups. They review issues and recommend activities of the TWGs. And the Steering Committee is composed of Dr. Nelia P. Maramba as the chair, Ms. Lilibet de los Reyes as co-chair, Maria Lourdes Santiago of Philippine FDA. She used to be a member of committee. Dr. Fabian Dairit, Professor Leticia Barbara Gutierrez, Dr. Constancio de Guzman, Dr. Francis Wade Gomez, Dr. Eleanor Roquel, and Mr. Emmanuel Velasco. Then we have the Technical Working Group 1, who are working on the general notices. Okay, they share the same, almost the same scope of work with the technical working group 2. It's just that they differ in the objective or the general work. Okay. So this is TWG1 is chaired by Dr. Benvenido Balotro and co-chaired by Ms. Pia Rose Bellarmino. Rounding up the committee or the group are Ms. Jocelyn Valderrama, Dr. Alicia P. Catabay, Dr. Grisebio Antonio Alejandro, Dr. Marilu Nicolas, Dr. Richard Nixon Gomez, and Mr. Philip Hus. Then we have Technical Working Group 2. Okay, as I've mentioned, because basically they have the same responsibilities, but their objective, their scope of work is more on testing and standardization. Okay, I'm chairing TWG2 with Mr. Michael Hunsay as co-chair. Then we also have Dr. Noel Kiming for chemistry, Dr. Jessica Ray for molecular biology, Professor Natividad Lakdan for botany, Ms. Leslie Lambio Rosas for agriculture, and Ms. Maria Lourdes Samonte from FDA. Okay, this slide now introduces you to the accomplishments of the project. First, we now have a criteria for inclusion of herbal medicines and medicinal plants in the pharmacopoeia. What are these criteria? The plant must be locally available and used by the community. It must have proven efficacy and safety. It must be registered as a single plant herbal medicines with dosage forms according to A0172 guidelines for regulation of herbal medicine. Then we have the 10 medicinal plants recommended by DOH, excluding nyog-nyogan because of safety issues. And then the next recommended 10 medicinal plants based on evidence for efficacy. So, the list consisted of herbal medicine, traditionally used medicinal plants, DOH recommended medicinal plants, and other medicinal plants with evidence of efficacy and safety. So what are initially included in this list of plants? We have Vitex negundo, Blumea balsamifera, Eresia microphylla, Menta villosa, Peperomia pellucida, Momordica charantia, Senna alata, Plecanthus amboynicus, Zingiber officinale, Allium sativum, Sidium guajava, Moringa oleifera, Leucaena leucocephala, Senna fistula, Curcuma longa, and Cocos nucifera. Then the contents of the general notices are being finalized. We have cultivated and harvested some plants and continued sourcing of plants. The harvested plants were also processed. Initial microscopic analysis and initial work for DNA barcoding have been done. Then the chemistry and pharmacy group have tested, are doing testing, standardization, and validation. We also have sample monographs. So, just like with other research projects, we have encountered difficulties, foremost of which is the prevailing conditions brought about by COVID-19, which affected the availability of plant materials and laboratories, reagents, supplies, and equipment. Primarily because of the very limited or no movement at all. The plants died and were therefore have to be replanted. Okay. Reagents and supplies cannot be delivered. Also the same with equipment, and the laboratories are not available, or uh, they were closed down. Okay, so what's next? What's next is what we're going to continue with the activities as planned, such as the laboratory work. Now that the laboratories have been opened, we also have to work on the approval of general notices and monographs, then the publication of the Philippine Herbal Pharmacopoeia, and finally its declaration and adoption as the official book of standards and references. Okay, and then the steering committee is preparing a list of plans for sustaining and institutionalizing the publication and updating of the Philippine Herbal Pharmacopoeia. I think that's my last slide, so I'll leave you with this quote by Tori Amos. Healing takes courage, and we all have courage, even if we have to dig a little to find it. Thank you.
So thank you, Professor Palakpak, for that talk. The link for the program catalog will be posted in the chat box for download, so please check that when you post it. To our participants, please feel free to write your questions in the Q&A box at the bottom of your screens. We now proceed to our next talk, Dr. Chun Tao Che, to discuss the key points of the WHO guidelines on good herbal processing practices for herbal medicines. Dr. Che is currently the Harry H. S. Fong Professor of Pharmacognosy at the University of Illinois at Chicago, UIC, and the Director of WHO Collaborating Center for Herbal Medicines. He has over 30 years of research and teaching experience in the USA and Hong Kong in the area of natural products and Chinese medicine. Specializing in phytochemistry and biological evaluation of secondary plant metabolites, he is also knowledgeable about the authentication, quality assessment, and ethnomedical applications of Chinese medicinal herbs. Dr. Che has published over 250 scientific papers on the following topics: isolation and structural determination of plant metabolites, standardization and quality of medicinal herbs, and toxicological activities of medicinal plants and botanical preparations, including pharmacokinetic analysis of herbal medicines. So please welcome Dr. Chun Tao Che.
Good evening, ladies and gentlemen. First of all, let me bring from Chicago my warmest greetings to every participant of this summit and wish you a happy, healthy, and prosperous new year. I would also like to thank the organizers for the invitation to participate in this summit. Back in 2014, I had the pleasure of participating in the drafting of one of the WHO Good Practices Guidelines for Herbal Medicines. That is the WHO Guidelines for Good Herbal Processing Practices. Today, I'll take a few minutes to make an overview of this piece of document and highlight its key points. For the sake of convenience, I'll refer this guideline as GHPP in this presentation, standing for Good Herbal Processing Practices. As you may know, prior to the publication of the GHPP guidelines in 2018, WHO has already published 2 good practices guidelines for herbal medicines, and they are the Good Agricultural and Collection Practices, GACP, published in 2003, covering the cultivation and collection of raw herbs. And then in 2007, WHO published the Good Manufacturing Practices, GMP, for herbal medicines, dealing with the manufacturing of final herbal products. But as you know, medicinal plants would undergo several treatment procedures in between, after being harvested and before going into the manufacture stage. This is where GHPP comes in. GHPP serves to link together the process of collection or harvest of raw herbs and the final manufacturing in the pharmaceutical factories. So what are the GHPP guidelines about? This is the table of contents of the document, and I'll quickly walk you through it. After fresh herbs are harvested or collected from the growing site, they will undergo a series of simple procedures, which are collectively called primary processes, to provide the herbal materials. In the example I use here, after harvesting the ginseng plant, the root part is sorted out, cleaned, and dried to give the herbal material, which is the raw ginseng root. What will then happen to the raw roots? They will go through a set of treatment processes, which are referred to as the secondary processes, to provide the herbal preparations. In the case of ginseng, the herbal preparation can be the cooked and dried roots, or it can be in the form of ground powders or liquid or dried extracts. These herbal preparations are then manufactured into the desirable products following GMP processes to provide ginseng tea bags, capsules, or other dosage forms. Now, let us take a closer look at each of the steps. The primary processes refers to some simple post-harvest procedures, usually performed at the harvest or collection site. In general, the raw herbs will be sorted to obtain the medicinal plant part, washed and cleaned, and finally dried to remove excessive water. The herbal material is then treated by the secondary processes. The secondary processes may include further cutting according to the traditional practice, grinding, or other treatments such as sweating, baking, steaming, or stir-frying. In some cases, the herbal materials are fumigated or irradiated for preservation purposes. And during the secondary processing, adjuvants are often used, such as the use of wine and vinegar when stir-frying the herbal material, or using honey during baking. In different traditional medicine systems, other adjuvants may also be used. Then why are the secondary processes needed? There are several good reasons. The secondary process may serve to improve the purity of the herbal material, prevent damage from molds and other microorganisms, detoxified intrinsic toxic ingredients, diminish undesirable side effects, modify therapeutic properties, or to enhance therapeutic efficacies. The exact purpose of performing secondary processes depends on the traditional practice and the properties of the individual herbs. In some specific cases, such as ginseng and some well-known toxic plants, such as aconitum and nux vomica, traditionally, they are required to be treated with a series of specific procedures tailor-made for that particular herb. These well-documented traditional procedures already laid down in the herbal literature are referred to as special processing in the GHPP guidelines. Now, in many cases, the herbal materials may be required to be further processed by physical methods such as extraction, distillation, fractionation, and purification in order to obtain cell-free preparations for downstream manufacturing of the final products. As a matter of fact, some of these preparations can be refined and directly consumed by the patients, such as in the form of decoction, infusion, fluid extract, and tincture, etc. Fermentation is another process that may be taken to prepare the fermented product. The final step of GHPP is to turn the herbal preparation into its final dosage form. Depending on what the final dosage form is, appropriate GMP processes are carried out in the manufacturing facilities to produce liquid dosage forms, such as fluid extract and decoction, or the solid dosage forms, such as powder extract or capsules, or other dosage forms, such as cream and plaster. Now, in the GHPP document, the last part covers other technical issues, such as processing facilities, packaging and labeling, storage and transportation, and equipment. It also addresses the QA/QC issues, documentation requirements, personnel training and qualifications, and ethical and legal considerations. The document also addresses the need for research training and information sharing, intellectual property rights and benefit sharing. And finally, the proper use of threatened and endangered species is also highlighted. Ladies and gentlemen, to conclude my talk, let me summarize the key concepts of GHPP. First, it recognized that the safety and QA/QC of herbal medicines at every stage of the production process are of prime importance. And GHPP is integrally linked to GACP and GMP. The primary goal of setting the GHPP guidelines is to provide general and specific technical guidance applied to the preparation of herbal materials, herbal preparations, and herbal dosage forms. It also provides a model for the formulation of national or regional GHPP related SOP. At the same time, the document contributes to the herbal QA/QC to promote the safety, efficacy, and sustainability of herbal medicines. The GHPP guidelines should be used with reference to the national or regional quality standards, and it must be carried out in accordance with the legal and environmental requirements, as well as the ethical codes or norms of the community and countries concerned. It is suggested that scientific research is needed to better understand and to provide evidence to support the traditional methods of herbal processing. And developing SOP for individual herbs and proper documentation are strongly encouraged. Finally, each country is urged to develop its own national guidelines and to establish their regulatory requirements for herbal medicines. Now, here is the website where you can find the GHPP guidelines. Ladies and gentlemen, due to time constraint, I can only give a brief overview of the GHPP guidelines today. Please take a look at the document itself if you are interested. If there is anything you may want to discuss, please drop me an email. I will be very happy to further discuss with you on this subject. Finally, thank you for your attention, and I wish you all the very best in 2021.
Thank you, Dr. Che, for sharing your expertise with us. Again, we'd like to remind everyone that for any questions that you might have during the presentations, please just type into our Q&A located at the bottom of your screen. Our next speaker is Leslie Angela Lambio Roses to discuss the good agricultural practices to ensure the best harvest. Professor Roses is an assistant professor at the University of the Philippines, Los Baños, and is also the project lead for the agricultural component of the National Integrated Research Program on Medicinal Plants. She finished her Master of Science in Agricultural and Horticultural Plant Breeding and BS in Agriculture from the University of the Philippines, Los Baños.
Hello, everyone. I am Assistant Professor Leslie Roses from the Institute of Crop Science, University of the Philippines, Los Baños. For the next 10 minutes or so, I am going to share with you good agricultural practices in medicinal plant production to ensure a better harvest. First of all, what are good agricultural practices, or GAP as we commonly call it? Good agricultural practices are, as defined by FAO, approaches that apply recommendations and available knowledge to addressing environmental, economic, and social sustainability for on-farm production and post-production processes, resulting in safe and quality food and non-food agricultural products. In the Philippines and ASEAN region, we have PhilGAP and ASEAN GAP respectively. But right now, both PhilGAP and ASEAN GAP has yet to release guidelines specific for medicinal plants. Fortunately, the World Health Organization have released GACP, or Good Agricultural and Collection Practices for medicinal plants that can be adopted by the rest of the world. The guidelines found in this document is the main reference of this presentation. So why is it important to adopt good agricultural practices in medicinal plant production? Following a standard set of protocols will help ensure sustainability of supply of raw materials to end users and ensure that the raw material is safe and of superior quality, which is of course our foremost priority in developing herbal medicines. GAP practitioners also enjoy the benefits of having higher productivity, profitability, safer and cleaner work environment, and market assurance, to name a few. Now let's proceed to the general guidelines in applying GAP in medicinal plant production. Say that we are to start a medicinal plant farm. Where should we begin? First, in selecting what to plant, we can refer to the National Pharmacopoeia or other authority for the choice of crop. Local and common names are unreliable bases of identity. It is absolutely essential that we correctly identify the plant we intend to cultivate. For example, we manage to obtain a specimen of our preferred plant. It is best to have the identity of that plant verified by professional taxonomists or authorities from the Museum of Natural History. Other traits that are useful for identification, such as physical, ecological, chemical, and genetic markers, should also be documented for authentication purposes. Next step, once the identity of our intended medicinal crop has been verified, it is time to secure good quality propagation materials. When obtaining propagules for field establishment, be it seeds or vegetative organs, the selected mother plant should be disease-free and preferably exhibits resistance and or tolerance to biotic and abiotic stresses, and also contaminant-free so as not to proliferate unhealthy plants in the farm. If one is acquiring planting materials from an external source, make sure the supplier is reputable and the planting materials are genuine and not substandard. Next, well, before field establishment, it is necessary to do site selection and evaluation to determine if the area is free from soil, air, and water hazards. Performing suitability analysis will determine if the existing physical factors like climatic and edaphic conditions or biological factors like presence of flora and fauna and sociological factors like security and market access are conducive for growing medicinal plants. Knowledge of historical land use and agricultural practices performed previously in the site and also in adjacent lands will also provide an insight on potential hazards that may exist. And this can be confirmed by conducting a complete soil analysis to check soil hazards like presence of heavy metals like lead, arsenic, and cadmium, in addition to other soil parameters that we need to know like soil fertility, pH, and others. Similarly, a water quality analysis of different irrigation sources and water bodies present in the site should also be checked for contaminants. If any of these hazards are present and not addressed, they will have a significant and negative impact on the quality and food safety of the plant material that may render it substandard and unmarketable. However, if the site succeeds in passing the suitability and is found to be free of any hazards, crop establishment and the management operations may now proceed. All inputs and activities should be recorded diligently, a hallmark feature of good agricultural practice. One of the benefits of strict record keeping is that if, say, a certain batch of raw materials was found to be substandard or unacceptable, the grower can trace back the root cause of the problem and may apply interventions accordingly. The recommended cultivation practices specific to each crop should be followed. In the absence of such, the traditional practice of production can be followed, and the optimization may be done later by a concerned agency. The type and timing of implementation of cultural practices depend on the plant's growth and development pattern. Special practices are also done for some crops to enhance their productivity. For example, in Ampalaya, shown in the photo, trellising is done to support its viney growth habit. Pinching is done during its vegetative stage to induce lateral branching to produce more leaves. Flowers are removed to reallocate the photosynthase towards the vegetative organs since it is the leaves that are the ones that are harvested for herbal medicine production. Another example of medicinal plant with specific growth requirement is the Olasimang Batno, which is also shown in the picture below. It needs to be grown under partial shade in order for it to be productive. Next is pest management, which is also important in ensuring high crop productivity. To maintain high quality profile of raw materials, diseased and damaged plant parts are completely disregarded or discarded. Integrated pest management or IPM is the recommended approach to pest control under good agricultural practice. Agrochemicals like chemical pesticides are used only as a last resort. But if it is inevitable, the use of agrochemicals should strictly fall within the maximum pesticide and herbicide residual limits set by national or regional guidelines. The next production aspect is soil and water management. Soil nutrient management will depend on the existing soil fertility gathered from the soil analysis conducted, and also the plant's specific nutrient requirement. Precision is key, as excessive fertilizer application, whether they are from organic or synthetic sources, can cause environmental problems like leaching that may lead to water pollution. As for irrigation, clean and contaminant-free water must be utilized to ensure both plant and human health. Soil and water conservation measures should be implemented in the farm to avoid erosion, waterlogging, and even flooding as seen in this photo. These stresses incur damage on plants, especially those that are waterlogging sensitive, and can adversely affect their yield performance. Next is harvesting. Harvesting should be done at the optimal season or peak time in the plant's life cycle. This is because the concentration of biologically active constituents varies with the stage of plant growth and development. Weather conditions at time of harvesting also significantly affects the quality of plant materials. It is not advisable to harvest on a rainy day or during humid days, as these conditions are conducive to harboring fungal pathogens that may damage the harvest. Also, proper hygiene of personnel and the tools used for harvesting should also be observed to prevent possible contamination. For example, tools and containers should be regularly sanitized, and harvested materials should not be placed on the ground. And finally, the last stage is the post-harvest processing. Post-production practices vary depending on the specific medicinal plant and how they will be utilized, whether as fresh material or as dried material. Harvested plant materials still go through post-harvest handling, such as inspection, cleaning, sorting, drying, if applicable, packaging, transporting, all within appropriate facilities that are up to standard. There are also some medicinal plant materials that require special processing practices to improve their purity or reduce their drying time, prevent further mechanical damage, detoxify indigenous toxins within them, and of course, enhance their therapeutic efficacy. Significant yield losses may also be caused by improper post harvest processing. Therefore, much care must also be observed during this stage of production. In summary, adoption of good agricultural practice in medicinal plant production ensures a sustainable production of safe and quality plant materials for the benefit of the end users and, of course, the herbal medicine consumers. It also ensures crop and farm productivity for the benefit of the growers, and also environmental and human health is ensured for the benefit of, well, practically all stakeholders involved and even beyond. And with that, I end my presentation on good agricultural practices to ensure a better harvest. Thank you all for listening.
Okay, thank you, Professor Lambi Roses, for that comprehensive presentation on good agricultural practices. We will now open and proceed to the open forum. So, to everyone, to our participants, please feel free to type in the Q&A chat box located at the bottom of your screens, or directly at the chat function of this webinar, and we will read them out, directed preferably to your speaker of choice, or open it up to our panel. Okay, so while waiting for our participants to share their questions in the chat box or in the Q&A, we'll proceed with Dr. Lazarte for a question.
Hi, thank you, Paolo. I think as of now, no, GAP and GHPP are not may not be as well known as the GMP, and it is not also legislated, I think, no. So, um, for Professor Che, uh, for us to develop a national GHPP, no? So where do we start? Who usually initiates this? Would be this from the regulators first, or would it be from the academe or institutes for us to start working on a national GHPP guideline?
Thank you for the question. Can you hear me?
Yes, sir. Very well.
Good. Good. Yeah, I think for both GAP and GHPP, it starts uh, by different sectors. Now, as far as I'm concerned, I think for any kind of these good practices guidelines, it requires some scientific support, scientific evidence to support whatever is proposed in the guideline. So in that respect, I think research institutes would be heavily involved. And eventually, of course, the guidelines should be adopted or approved by some controlling agencies or policy agencies. And at that stage, I think the regulatory agencies would be involved. So far as I think all these guidelines are mostly only recommendations, and they are not really put under have any legality or legal implications at this time, as far as I know. Yeah.
Yeah, because the GMP is part already of
Yes.
regulation, really. But do you see like a trend going this way, that could it possibly, or should it be, or should it just be a general guideline?
Well, personally, I think they should be. But of course, then in reality, I think the agencies or the regulatory agencies have to work closely with the industry to make sure that the industry can fulfill all these requirements. And so this is a process, I think, involved in developing this into legal status.
Yeah, similar to the pharmacopoeia, wherein we also involve the industry to see what can they do. I mean, we might have all the regulations, but if it's not practical for the industry to
Right.
do, then it's also not helpful. Thank you so much, Professor.
Thank you.
Okay, so we have two questions directed. We'll be addressing them in order. Directed to Professor Roses. We have one question from Mr. Gabuna, an organic farmer. Where can they find or have access to any advice or guidance, especially for organic farmers? That's for our first question for Professor Roses. And then we'll follow it up with another slightly related question.
Okay, so for advice on any crop related or any agricultural related question on farming, we can always go first to our local municipal agricultural office. So meron po tayo ng bawat munisipyo natin. So meron pong agricultural office. So they can, there, the agricultural officers, the agricultural technician, if you're interested in a gap certification of your farm or any particular commodity, they can link us with extension agents or employees from Bureau of Plant Industry or for Philippine National Standards. So yun po. So yun ang ating front line. We can go to the our respective municipal agricultural offices po. And then next.
Yes, we have our next question, Prof. Thank you for that. Are there any herbariums or farms here in the Philippines that follow the good agricultural practices? Could it possibly from the Bureau of Plant Industry, or I think they're asking for a specific area or location?
Okay. So there are many registered na herbal farms or farms that are cultivating herbal plants. So actually, our team is collecting from different farms all over the country. So, but as for if they are practicing GAP or if they are GAP certified, I do not know, but they should be. Kasi here in the Philippines, hindi tayo required, voluntary ang mag-GAP certify sa ngayon. And wala nga tayong GAP talaga for medicinal plants. So ang pina-follow natin ay for herbal or for medicinal plants, we can follow the fruits and vegetable code from PhilGAP. So as for if they are certified by PhilGAP, so wala po akong list, but kung meron po tayo, madami po tayong mga farms that are cultivating different medicinal plants all over the country.
Okay, thank you, Prof. Actually, there's a related question here from Josephine Kipe, who pointed out that their farm or by the Department of Agriculture in Region 8 is actually GAP certified already. I think it's related to your question about to what you raised about the current GAP standards for medicinal plants. They ask about if this qualifies, if they're GAP certified, if this qualifies to produce and process medicinal plants already.
Okay, so the We have a separate gap for I mean a protocol or guidelines for agricultural practices, and we have a separate then protocol and guidelines or certification for yung processing na. So yun po yung minamention kanina ni Ma'am na good management practices. So parang in this case kailangan na yung facility, kasama na rin yung facilities. Although kapag yung mga farms ngayon ay buo na, parang from kung halimbawa hindi lang sila producer, diretso na sa processing, ay pareho na silang GAP certified and GMP certified. So, kung, ano po ulit yung question? Sorry.
The question po nila is, if they're already GAP certified by the Department of Agriculture, does this also qualify them to process and produce medicinal plants already?
Okay. So, yun nga po, parang depende po dun sa kailangan specific po kasi yung magiging requirements or standard set for herbal medicine production na po. Iba po yung from yung herbal materials, yung gap na may safe ka na herbal materials, iba na po yung parang kung makakapasa siya dun sa standards na set nung doon na ng pharmacy side, ganyan. Kung wala siyang mga contaminants, etc. So, kung GAP certified, most likely you need the extra step for having your herbal materials really food safe.
Okay, thank you, Prof. Now we'll also raise several questions to Professor Palakpak. There were mentions of, there's an inquiry about if the herbal preparation, we have from Renzi Hierro, they're asking if the herbal preparation that they're currently working on could be considered for the pharmacopoeia since it consists up to 10 herbal plants. Although they did not specify which plants are these specifically, they are in doubt because it was mentioned earlier that an herbal preparation should be made up only of single plants.
Hello, Paulo?
Yes po.
Okay. Naglabas po ng guidelines ang steering committee para po dun sa mga plants na pwedeng idagdag dun sa listahan ng listahan para sa Philippine Herbal Pharmacopoeia. Pwede pang ipadala siguro sa kanila yung yung criteria kung gusto nila.
Okay.
Pero at the moment po, single plants, hindi po mixture.
Okay. So again, it might need further inquiry, but for now, single plants lang po. Prof, also related to that is, this is a question from Ping Peria from General Santos City, a policy researcher. How do you think should AO 172 be amended given greater recognition of indigenous people's traditional knowledge of their indigenous medicines? Since noted that our own pharmacopoeia is circa 2004 and needs to be accommodating new developments and updated.
Okay, AO 172 is intended for the registration of herbal medicines. Yung, as for the recognition of indigenous knowledge and the rights of indigenous people, I think may ginagawa na dito yung commission. I just forgot ano yung tawag doon sa commission na yun.
Okay, thank you, Prof. Okay, we have also additional inputs from our panelists.
I think recognition would be more of the assigning also the intellectual property, no? So a lot of, I think it's not in the law, where is it? That those coming from indigenous peoples, no, the IP should also be shared with them. No? So there is also, but I don't think it's in the AO 172, it's in another law. So recognition also really should be given to these indigenous peoples if this plant is really endemic to them only. No, so they really should be recognized. The IP should be shared with them. At the same time, royalties or earnings or whatever should also be shared with these IPs. Thank you, Palo.
Okay, thank you everyone for your responses. We'll entertain one last question. Um, so I guess this is for everyone. Uh, is there okay? We have a question from Nico Buhay. Is there a particular reference or website that you could advise students to read on and study for Philippine herbal plant related researches? This is addressed to anyone from the panel. As well as if there are any unanswered questions, we'll be addressing these and forwarding them to our panelists so that they can be answered or at least addressed in the chat box. Thank you.
Okay, if no one wants to take that question, there are some databases. The Herdin database of PCHRD, there are a lot of studies from different academic institutions that are housed there, mostly the abstracts, but you can also ask for help to get original articles on these herbal researches. What was the other database? The Herb. I think in Pitak, they also have an herbal database where you can look at. You can come over to the Institute. We have some materials here if you want to look at them also. And I'm sure Professor Check, would you like to add to?
Yeah, our department has built a database for natural products for many years, and it is known as the NAPRAlert, standing for Natural Products Alert. And it was started by the late Professor Norm Farnsworth. And so if any one of you are interested in getting information from the NAPRAlert, yeah, please just you can just Google NAPRAlert and you'll get the website, and we'll be happy to connect with any one of you who are interested. Thank you.
Right, thank you. Thank you to our panel for sharing with us their expertise. And I would like to proceed to awarding them of certificates. So we'll be presenting them with our certificates. The citation reads, the Institute of Herbal Medicine, National Institutes of Health, University of the Philippines, Manila, presents this certificate of appreciation to our speakers, Professor Jocelyn Bautista Palakpak, Dr. Chun Tao Che, Professor Leslie Angela Lambio Roses, in recognition of their invaluable contribution as speakers at the 3rd Philippine Herbal Medicine Summit with the theme Ensuring Quality of Herbal Medicines Through Research, Regulation, and Cooperation, held today on January 25 through 27, 2021. So again, we thank all our speakers for sharing with us their time and their expertise.
Thank you for having me.
Thank you so much.
Thank you.
Thank you also to our participants for their questions. We'll now proceed to the updates on herbal therapies for COVID-19. And for this, again, just type in your questions at the Q&A box throughout their presentations. And then we'll be collecting them and addressing them at the open forum at the end. So now we proceed with our next talk is to be given by Dr. Philip Ian Padilla to discuss the updates on Tawa Tawa. Dr. Padilla is currently the Vice Chancellor for Academic Affairs of the University of the Philippines, Visayas. He is a professor of microbiology and former UP faculty regent. He was also the former chair of the Division of Biological Sciences, College of Arts and Sciences, and former director of the National Institute of Molecular Biology and Biotechnology at the UP Visayas. He was a graduate of UP Visayas BS Biology cum laude, UP College of Medicine, and Nagasaki University's Institute of Tropical Medicine for his PhD in Medical Science and Bacteriology. He was trained as a postdoctoral research fellow in biochemistry and cell biology at the Pulmonary Cell Care Medicine Branch, National Heart, Lung and Blood Institute at the National Institute of Health, Bethesda, Maryland. His expertise is in molecular biology, bacteriology, and public health. Let's welcome Dr. Padilla.
Hello, good morning. So thank you for the invitation to speak to you this morning. This is our study on the randomized controlled clinical trial on the efficacy and safety of tawa-tawa, which is Euphorbia hirta, extract as an adjunctive treatment for mild to moderate COVID-19 patients. I am the principal investigator, P. Padilla, and our co-investigators are the former secretary of the DOH, Dr. Pauline Obial, Dr. Jubert Benedicto from the Internal Medicine Department of PGH, Dr. Luz Vicente Guanzon from Corazon Luxin Memorial Hospital in Bacolod City, and Dr. J.C. Malabad of UP-PGH, UP College of Medicine. The implementing agency is the University of the Philippines, Visayas, and trial sites are the UP Philippine General Hospital, the Quezon Institute Mild COVID-19 Isolation Facility, and the Corazon Luxin Montelibadong Memorial Regional Hospital in Bacolod City, and other quarantine facilities with mild COVID-19 patients are also included in Bacolod City and Negros Occidental. Based on previous studies on the oral toxicity and other pharmacologic effects of tawa-tawa, I just summarized here the different studies, the different animal studies that have been published or have been in literature for the past 5 years. I won't go through them one by one. This is just the first page. But overall, the LD50 and the other adverse effects have been recorded at a particular dose, and more or less there were no biochemical or mortality that were recorded during these particular experiments. Phase 1 clinical study for tawa-tawa has already been published in the Philippine Journal of Experimental and Clinical Pharmacology. It is the phase 1 safety and dose escalation trial of orally administered tawa-tawa capsules in healthy Filipino subjects under fasting conditions. It was randomized with 5 cohorts of 3 subjects each, with increasing doses of tawa-tawa with 1,000 milligrams to a maximum of 24,000 milligrams. The results showed that the maximum dose that had no effect was about 24,000 milligrams, with the dose-limiting toxicity reported in 1 out of 6 subjects who were given this dose. It was also associated with increased hemoglobin count by more than 16 grams per liter. There were no abnormal changes that were recorded in the ECG, in the CBC, platelet, bleeding parameters, blood chemistry, and other vital signs that can be classified as dose-limiting toxicity. Only 1 report of clinical toxicity of flatulence was reported in the 12,000 milligram cohort. So overall, the maximum dose for tawa-tawa has been recorded at 24,000 milligrams, and there's a 90% confidence interval. There is a 90% confidence that the true probability of experiencing a DLT will not exceed at 32%. I also like to cite the management of thrombocytopenia and flu-like symptoms in dengue patients with herbal water of Euphorbia hirta. Thus, we came up with the possibility that Euphorbia hirta or tawa-tawa might also be effective in other viral diseases such as COVID-19. The general objective of this study is for the safety and efficacy of the standard aqueous formulation of Euphorbia. It's a spray-dried extract previously developed under DOST PACHRD 2+Lunas program as adjunctive treatment for mild to moderate cases of COVID-19. The specific objectives is to find out the optimal dose of the standard aqueous tawa-tawa STE. We'd also like to determine if the supplementation has ameliorative effects on the different clinical signs and symptoms of COVID-19 patients. We would look at the different hematological and biochemical processes of patients with mild COVID-19, if that has an effect as well. To determine also if we could have prevent the progress of patients with mild symptoms to more severe conditions, and determine if there are adverse effects in patients with COVID-19 taking the supplement. The study design is an interventional randomized controlled phase 2 dose finding study design. So the study sample where we recruit patients, they will be randomized into three arms: standard of care plus two caps of tawa-tawa 3 times daily, and standard of care 3 caps of Euforbex 3 times daily. So this is, we computed the sample size. So the total for control standard of care is 50 mild and 40 patients for moderate. For treatment 1 and treatment 2, the same ratio has been prescribed for the different arms. Inclusion criteria are patients with 18 to 59 years old. They were hospitalized and confirmed with the WHO criteria with a positive PCR in the specimen that was submitted, and that fulfill also the mild to moderate COVID-19 case definitions by the WHO interim guidance on clinical management of COVID-19. So these are the case definitions. I won't go into them already. So exclusion criteria: those with a history of allergic reactions to euphorbia, suspected ongoing or active or ongoing infectious disease, pregnant or breastfeeding women, severe or critical COVID-19 by case definition, participants who are also undergoing other clinical trials, patients undertaking aspirin, those with hypertension and diabetes mellitus, patients with disturbances in their liver function test, and any other medical conditions that might affect the test for COVID-19 patients. Anybody can withdraw during the trial if there's progression of disease from severe to critical case, if the liver function tests escalate, and development of hypersensitivity reactions to the extracts. And of course, other conditions that will indicate abnormality in the laboratory as well as the clinical signs and symptoms. This is the flow chart for the admission of COVID-19 facility. This is the randomization and the primary outcome of the patients that will enroll in this particular program. These are the secondary outcomes. We want to look at the improvement of 2 categories relative to the baseline on a 7-category original scale of clinical status. Time for clinical failure, time for the resolution of symptoms, time to normalization of the different biochemical and hematological parameters, the time to hospital discharge, the duration of time on supplemental oxygen, percentage of patients with adverse events, and the time to RT-PCR virus negativity, and the proportion of participants with post-treatment infection. We would like also to tell you that the clinical trial was supposed to start last September 2020, but because of different factors, we have not been able to accomplish the recruitment of the patients for this clinical trial. The funds haven't been downloaded yet, so UPV was able to give the bridging funds. The review for the FDA and the other ethics institutional review board is already available pending the payment of the registration, pending the payment for the review to the FDA. So by this week, we are supposed to be on target to remit the payment so that the IRB review results will be released by the FDA. There's already a MOA signed between UPV and DOST-PCHRD. Line item budget has already been approved. We have already hired admin and research staff, both for UPV and Corazon Doxin. The UP Manila Technical Leader Review have approved the research protocol information. DOHSJ Rep initial submission has also been submitted. So we are just waiting for the FDA IRB results. Additional partner hospitals and quarantine facilities in Negros Occidental have also been identified, and the letter of intent has already been sent to the governor of Negros Occidental. PPMP has been approved by UPB for the obligation of funds, and the initial bridging fund of around 84,000 has been approved by the UPB Chancellor for the review fees. There are still various procurement issues that we are encountering because of the bureaucratic red tape. Hopefully, this will be done by this week. So I guess that's it. Thank you very much. And again, thank you for the invitation to speak to you today about the Tawa-Tawa clinical trial.
you, Dr. Padilla, for sharing with us information about the Tawa-Tawa trial. Now we proceed to our next speaker, Dr. Fabian Dairit, to discuss updates on virgin coconut oil. Dr. Fabian M. Dairit is a full professor at the Department of Chemistry at the Ateneo de Manila University, where he has been teaching since 1983. He obtained his BS chemistry degree from the Ateneo in 1975 and his MA and PhD degrees in chemistry from Princeton University in 1978 and in 1981, respectively. He worked as a postdoctoral research associate at the Oxford University from 1980 to 1982. Dr. Dairit did the phytochemical work for the development of Lagundi, Vitex Negundo, as part of the National Integrated Research Program on Medicinal Plants. He has published over 50 papers in research journals, mainly in the areas of natural products chemistry, environmental chemistry, and coconut oil. In 2013, he wrote the popular book Coconut Oil from Diet to Therapy, and in 2015 to 2016, he co-edited the Encyclopedia of Common Medicinal Plants of the Philippines, Volumes 1 and 2, which are publications of the Philippine Institute for Traditional and Alternative Medicine.
Good evening to everyone. First of all, I would like to thank Dr. Cecilia Maramba Lazarte for this invitation to talk at this third Philippine Herbal Medicine Summit to share updates on the use of virgin coconut oil against COVID-19. This talk is entitled Virgin Coconut Oil, a Multifunctional Food Against a Multifaceted Disease. This talk is in two parts. First, COVID-19 is a multifaceted disease. I will share a description of the recent challenges of COVID-19 to provide a rationale for the use of virgin coconut oil. Second, VCO is a multifunctional food. I will describe updates on the use of VCO which may address these recent challenges of COVID-19. I would like to share with you a bit of history. On January 29, 2020, during the very early stages of this pandemic, Dr. Mary Newport and I wrote a paper which we posted on the internet suggesting the use of coconut oil against the novel coronavirus, which was then still called 2019-nCoV. While some of the initial analysis that we made last year has changed, the proposal to use VCO against the disease remains valid and may even be more urgent. There's this famous saying, when all else fails, read the instructions. We should apply this to the challenge that we have with COVID-19. When all else fails, read the RNA. Of course, the RNA contains the instructions for this coronavirus, and that is what is giving us problems. In the first part, I would like to discuss how COVID-19 is a multifaceted disease. Severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, is the virus that causes the COVID-19 disease. It is an RNA virus with instructions for how it can infect and damage the cell, as well as replicate itself. It is a small virus, a nanoparticle of about 80 to 120 nanometers, with a spike protein that enables it to attach to the ACE2 receptors of the body cells and release the viral RNA into the host cell. This virus, SARS-CoV-2, is a lipid membrane which can be inactivated by surface active agents or surfactants. This is the basis for the recommendation to wash your hands often with soap and water. This lipid membrane does not mutate, unlike proteins. This drawing shows how SARS-CoV-2 attaches to cells through the ACE2 receptor. The lipid membrane is a double layer. and it is believed that its components are derived from the membrane of the cell from which it exited. The spike protein, which we shall describe later, attaches to the ACE2 protein. The angiotensin converting enzyme is an enzyme attached to the surface of cells located in the lungs, the heart, the kidney, intestines, arteries, and many other parts of the body. ACE2 lowers blood pressure by catalyzing the hydrolysis of angiotensin, a vasoconstrictor peptide, into angiotensin, a vasodilator peptides. The amount of ACE2 tends to increase as we age, and this is the reason for the higher susceptibility of older people to this disease. Much research has been focused on the spike protein, which includes development of vaccines, search for drugs, and development of monoclonal antibodies. On the other hand, the lipid membrane has not been targeted, much less studied. COVID-19 is a systemic disease. Here I will briefly trace the sequence of events from viral infection to disease. First, the spike protein of SARS-CoV-2 binds to the ACE2 receptor, which makes this virus more infective than previous viruses. The target, the ACE2 receptors, are unfortunately found in numerous parts of the body, so there are many targets that this virus can enter. Although COVID-19 was originally thought to be only a respiratory disease, the virus attacks many other cells and organs, causing many other medical conditions. When the immune system over-responds to the viral attack, a cytokine storm occurs, which can damage the body. In addition, long-term effects are now recognized as part of COVID-19, or the so-called long haulers. This coronavirus can lodge in many organs in the body and stay there for a long time. This photo shows particles of the virus in the intestinal cells of a recovered patient 92 days after the onset of symptoms. The S1 protein can break off and cause widespread inflammation in many organs of the body. Here are two reports which can be explained by the hypothesis that the S1 protein of this virus can be detached, and this can cause inflammation, including inside the brain. Therefore, the secondary effects of inflammation may sometimes not be due to the intact virus, but due to parts of the virus that have broken off. What has been the pharmaceutical response to COVID-19? The strategies against COVID-19 can be classified into three types. First, we have development of antiviral drugs. In principle, this includes development of novel drugs. However, because developing novel drugs from scratch can take up to 10 years at great cost and low success rate, all of the antiviral drugs that are being tested are repurposed drugs. Second, improving the response of the immune system through vaccines, monoclonal antibodies, or convalescent plasma. This has been where most of the effort has been. The third is to develop anti-inflammatory drugs, such as anti-inflammatory agents, such as drugs and convalescent plasma. This early study in September 2020 on a mutation in the spike protein showed how the infectivity of this virus increases with the mutation of aspartic acid to glycine on position 614. Here we can clearly see the importance of being able to read the instructions, the viral RNA. Since then, numerous other amino acid mutations in the spike protein alone have been detected. As early as September 2020 also, mutations in the spike protein have been reported in different regions in the world. This may mean that the efficacy of vaccines may vary depending on the variant against which the vaccine was developed. Mutation produces SARS-CoV-2 variants. However, an antibody which is neutralizing against one variant may be non-neutralizing against another. Mutation may lead to a condition called antibody-dependent enhancement. In ADE, a non-neutralizing antibody binds the variant coronavirus particle, and this enhances the entry of this variant into the cell. This has been described as a Trojan horse effect. The formation of large complexes of virus particles held together with non-neutralizing antibodies may cause inflammation and airway disruption. So the strategies against COVID-19 are threatened by the occurrence of mutations, the threat of ADE, antibody-dependent enhancement, and long-term inflammation from COVID-19. So what can VCO do? First of all, I prefer to consider VCO as a functional food, since it is a food, and maybe as an herbal medicine later. Just as a little background on virgin coconut oil. Virgin coconut oil is obtained directly from fresh coconut meat without chemical processing. Fresh coconut meat itself contains about 35% coconut oil. So if you take coconut meat, about 1/3 of that is coconut oil. And VCO is generally recognized as safe or GRAS. The major fatty acid of coconut oil is lauric acid, which makes up about half of coconut oil. Coconut oil is a unique oil since it is the only commonly consumed oil which is about 2/3 medium chain fatty acids. All other fats and oils are mainly long chain fatty acids. And the fatty acids present in the oil comes as a mixture of triglycerides. And coconut oil is a characteristic triglyceride profile. So how does coconut oil become antiviral? The original oil is made up of triglycerides, which are not themselves active. Lipase enzymes, which are present in many parts of the body, such as saliva, skin, digestive system, the liver, blood vessels, and others, hydrolyze the triglycerides and release the fatty acids. Trilaurin represents a major coconut oil molecule. Hydrolysis of trilaurin releases monolaurin and eventually 3 molecules of lauric acid. Very importantly, monolaurin and lauric acid can behave as surfactants. This figure describes the various steps in the viral life cycle, starting with attachment and ending with release of more virus particles. Previous work has shown that components of coconut oil can act outside the cell. So outside the cell, that is before the virus is able to enter the cell, monolaurin is able to disrupt the lipid membrane of the the lipid-coated viruses. The coronavirus family is a lipid-coated family. Other previous work has also shown that lauric acid can act inside the cell. This study done in 2001 concluded that lauric acid is able to inhibit the maturation of the Junin virus. Now I'd like to share with you the results of our work, which was funded by PCHRD DUST. And this work was done as a service contract in Singapore. 2 in vitro experiments were performed. The first experiment was designed to determine whether the compounds from coconut oil, that is lauric acid and monolaurin, had direct virucidal effect. That is, whether it is able to destroy the virus outside the cell. The results showed that lauric acid and monolaurin had virucidal activity at low virus concentration, but not at high virus concentration. This is readily explained almost as a chemical reaction, where if you had a certain proportion of lauric acid and monolaurin against a certain amount of virus particles, it is sufficient to break the membrane in the virus particles. But if you have a lot of virus particles, then the amount of monolaurin or lauric acid may not be sufficient. The second set of experiments sought to determine whether lauric acid or monolaurin can block or alter any process when the virus is already inside the cell. The results were surprising. The dotted red line at the bottom shows the expected result if there is inhibition of viral replication inside the cell. The solid pink line in the middle is the control with no added agent or reagent. The virus proliferates, but then kills the cell after about 24 hours. So the virus concentration also drops. What we observed was a solid blue line, where the concentration of the virus did not fall, implying that the cell survived. This result of enhanced cell survival should be studied further. This is a summary of our in vitro results. First, outside the cell, there's virucidal activity at low viral load. Second, inside the cell, there's enhanced cell survival. What is important to note is that neither mechanism acts on the viral proteins. Therefore, this may work, hopefully, regardless of mutation. This is a big hypothesis that needs to be proved. Now, I would like to share with you the results of the first ever double-blind clinical study on the use of VCO on suspect and probable cases of COVID-19. This work was led by Dr. Imelda Agdapa of the Food and Nutrition Research Institute and was funded by PCHRD DUST in collaboration with DUST Region 4A, the Philippine Coconut Authority, and the City of Santa Rosa. I will describe two of the indicators that were used. First, occurrence of signs and symptoms of COVID-19, and second, level of C-reactive proteins in the blood. The first result showed that recovery from COVID-19 related symptoms was more rapid in the VCO group versus the control group. Interestingly, immediately after the first dose of VCO, which was at a rather low level during day 1, 9 out of the 29 subjects, or 17% in the VCO group, already manifested improvement, compared with only 1 out of 28 subjects, or 4%, in the control group. Perhaps a more objective measure of inflammation is the C-reactive protein. The results showed that the CRP dropped much more rapidly in the VCO group versus the control group. By day 14, the mean CRP level in the VCO group was at about 3 milligrams per liter, and this continued to drop until day 28. In comparison, the control group stayed at the borderline inflammatory level of 5 milligrams per liter until day 28. VCO can be claimed to be a functional food. There is sufficient literature, which includes animal and clinical studies, that support its recognition as a functional food. What is evidence for the use of VCO as a functional food against COVID-19? The antiviral properties of VCO are supported by previous studies reported in the literature, as well as our own in vitro study against SARS-CoV-2. But certainly, more work on this needs to be done. The evidence for the immunomodulatory effects of VCO come from the literature. This is a topic that we should pursue. The results from the literature, as well as the first clinical study, support the claim that VCO is anti-inflammatory. So here you can see that indeed, VCO can be considered as a multifunctional food against COVID-19. The use of VCO has increasing significance in the light of recent reports on the effects of this virus. First, there's a continued presence of the coronavirus in various organs of the body, even after many days of post-infection, as shown in the study of detecting the virus in the intestinal cells. If one continues to take VCO even after infection, hopefully it will be able to address this problem of the continued presence of the coronavirus in the body. Second, the long-term inflammatory effects of this virus from detachment of its proteins can be addressed by taking VCO, which is anti-inflammatory. Third, although VCO may have virucidal effects, its more important effect may be its ability to enhance cell survival. And finally, it is possible, but remains to be proven, that the effects of VCO may not be affected by mutations of the virus. This will truly be a major contribution to the management of COVID-19. So with that, I will be very happy to answer your questions. And hopefully I've been able to convince you that virgin coconut oil is a multifunctional food against this multifaceted disease. So thank you for your attention.
Thank you, Dr. David, for sharing your results on coconut oil. Now for our next speaker, we have Dr. Cecilia Maramba-Lazarte to discuss the updates on Lagundi. Dr. Maramba-Lazarte is a professor of pharmacology and clinical professor of pediatrics at the UP Manila College of Medicine. She has a medical degree from the University of the Philippines Manila College of Medicine and has a Master of Science degree in infectious disease and a Master of Science degree in clinical trials, both from London University School of Tropical Medicine, University of London. She is currently the director of the Institute of Herbal Medicine, National Institutes of Health, UP Manila.
It is my honor and pleasure to present to you tonight some preliminary data from the Lagundi trial for COVID-19. So for the next 15 minutes, we'll be talking about some modern medicines from plants for infectious indications, a brief overview of COVID-19 so we would be able to understand the rationale for Lagundi use in this particular disease entity. We'll also have a backgrounder on what the other studies on Lagundi have been. And we will also look at an overview of the trial and some interim results. As we all know, plants produce structurally diverse compounds, and many of these compounds may have specific roles, including defense against phytopathogens. And sometimes these compounds are also active against human pathogens. And there have been several modern medicines that were derived from plants. We all know about quinine, which we use for severe malaria in combination with other anti-malarials. Even oseltamivir, which is used for the treatment and prevention of influenza A and B, is actually derived from your star anise, and it inhibits neuraminidase. And of course, the Nobel Prize winner for medicine in 2015 by Yu Tu was won because she was the one who isolated artemisinin from your Artemisia annua, which is the treatment of choice in combination with lumefantrine to treat your chloroquine-resistant Plasmodium falciparum. And we know that this COVID-19 affects our respiratory tract, both the upper respiratory tract, wherein you could have these mild symptoms, and if it already involves your lower respiratory tract, we would also have symptoms of pneumonia. Okay, so these are the different stages of infection of COVID-19. The first stage is your early infection or your viremic phase, wherein you have the fast replication of the virus in the patient. At this early stage, the patient has only mild symptoms, no? As we described earlier, some cough, diarrhea, fever, and headache. And their laboratory parameters are really not that deranged yet, no? Unfortunately, if they progress to stage 2, which is the pulmonary phase, okay, this would be the time when they would already have pneumonia, and they would have symptoms of pneumonia such as shortness of breath, tachypnea, having low oxygen levels or hypoxia, and if a X-ray or CT scan would be taken, they would already have abnormal chest findings, have some derangement in their liver function test, okay? And of course, they would need to be already treated in the hospital at this stage. For stage 3, wherein there is a severe host inflammatory response, no? So this is where the patient could already have acute respiratory distress syndrome, shock, cardiac failure. Oftentimes, this patient already has to be intubated. Their inflammatory markers such as the CRP, procalcitonin, troponin, D-dimer would be very much elevated, no? So this is really the critical stage of the patient. Now, if you look at the bottom row, it shows the potential therapies. And unfortunately, at the moment, the only drug that has been seen to decrease mortality is your steroids, no? Your dexamethasone. The other drugs have not yet really been proven to decrease mortality, especially in patients who reach stage 3. So this is also a illustration of the different potential therapies and what their targets are in terms of the life cycle of your SARS-CoV-2. So if you see some familiar names here, some of them have not panned out to be as helpful as we thought they could be, like your hydroxychloroquine and chloroquine, your lopinavir. They did not turn out as good as we thought they would be. Even your remdesivir, no, the WHO came out with a statement that it was not helpful in decreasing your mortality. So I just wanted to point out that your favipiravir and your remdesivir targets are your RNA-dependent RNA polymerase, no? So if you inhibit your RNA-dependent RNA polymerase, you would decrease your RNA synthesis, and it would also decrease replication of your SARS-CoV-2 virus. Okay, so now let's go to your lagundi, your Vitex negundo. So this is what your lagundi looks like. It is also called the 5-leaf chase tree. This is not marijuana, but it similarly has the middle leaflet, which is longer than the lateral leaflets. And our own Filipino traditional healers used it for cough and respiratory ailments in their patients. Now, I'm just going to show a few of the preclinical or non-clinical studies, meaning non-human studies, either in vitro or in vivo. In this particular slide, it shows the safety studies that Lagundi does not damage the DNA of cells. It has a very wide safety margin with its oral LD50 being very high at 114.95 grams per kilo in mice. And the chronic toxicity studies showed that indeed it was very safe. So the other non-clinical studies which were evidence of its pharmacologic activities included the cat trachea model, which showed that Lagundi had bronchodilating activity and smooth muscle activity. It also is a histamine inhibitor, and the bioassay which wherein they used the rat duodenum showed that it inhibited smooth muscle, and it also, which was used as a surrogate for bronchodilation. Other animal models showed that it had analgesic and fever-lowering effect, as well as an anti-inflammatory effect shown both in vitro and in the paw edema model. Now, what did the clinical trials show? So I just giving you a snapshot. The same studies were also done in adults. These are the ones done in children. No, so this showed the PEFR, no, which shows its bronchodilating potential at the end of therapy, which showed that indeed it favored Lagundi, wherein it had good bronchodilating activity in several clinical trials shown here. In terms of decreasing the frequency of cough in children using the syrup, no, it really showed that it had that antitussive effect, possibly due to its bronchodilating effect, that it reduced the frequency of cough in children. And in the usual viral cough, which usually lasts around 7 days, it reduced the cough to around 3 days only. Now, what were some of the studies done in relation to SARS-CoV-2? Actually, the data is not that much, but some in silico research showed that two of its compounds, your quercetin and your vitexin, showed a high affinity to also your RNA-dependent RNA polymerase, similar to that of remdesivir, no? Showing a potential antiviral activity by possibly inhibiting this RNA-dependent RNA polymerase. Now, vitexin, which is found in Vitex necundo, was also found to have a high affinity to 3 virulence proteins or factors, no? Your non-structural protein 1, your non-structural protein 3C, and your SARS-CoV open reading frame 7A. Although they have different mechanisms for its virulence, the vitexin had a high affinity to all 3 of these virulence factors, showing a potential to inhibit these virulence factors. Now, many studies have looked into the different compounds or constituents of Lagundi and finding out, no, what are the effects of these particular compounds. Several iridoids were indeed found to have bronchodilating effect, your agnoside, lagundin, and others. Some flavonoids were also found to be bronchodilating, luteolin, crisoplenol D, isoorientin. Some compounds or iridoids were anti-inflammatory in action, your lagundinin, some flavonoids, casticin, crisoplenol D, okay, and other effects. And the ones I mentioned a while ago, the vitexin and quercetin, having possible antiviral effects for SARS-CoV-2 itself and for influenza. So where do we see the role of Lagundi in the treatment of COVID-19? No? So this combination of compounds may actually be synergistic or targeting different aspects of the COVID-19 disease. It looks like it would be helpful for the symptomatic treatment of COVID-19, being an antipyretic, being a bronchodilator, and being anti-inflammatory. So its symptoms would be decreased because of the different constituents. But its possible antiviral compounds, no, would hopefully also decrease the replication of the SARS-CoV-2, no, so that we're envisioning that it should be given very early in the disease, no, in stage 1, while the patient still has mild signs and symptoms, so that they will not progress to moderate or severe disease. So this is our hope that Lagundi would even help decrease morbidity and mortality by stopping the progression, so that people infected will not go into stage 2 or stage 3 disease anymore. Now, what particular Lagundi product are we talking about? We're talking specifically about the near prompt IHM product. No? So this is the tablet, the 300 and 600 milligram tablet, plus the syrup, which is 300 milligram per 5 ml and 600 milligram per 5 ml. So these are registered herbal medicines. No? They have an HMR number. They are not supplements. They are herbal medicines. No? And this has been licensed out to several drug companies, and these are non-exclusive licenses. So these include your Ascoff Lagundi tablet and syrup, your Plemex tablet and syrup, and your Lagundex tablet. And there are actually several more brands in the market. So this is it. These are the different. So you notice it is tablet, no? It is not a capsule. Because early on when they were developing this particular formulation, when they tried the capsule, it had very poor dissolution time and poor absorption. And actually there are some Lagundi capsule products out there, and I've heard some of the patients complain that they've even excuse me for saying, pooped out the whole capsule, no? Kasi it was not absorbed properly, no? So we cannot vouch for the other Lagundi products. We can only vouch for those which are licensed by UP Manila, which are the tablet and the syrup. Let's go to our clinical trial. So the long version of the trial is called a 2-stage randomized double-blind placebo-controlled clinical trial on the efficacy and safety of Lagundi tablet and syrup, in particular the near prompt formulation plus standard of care compared to placebo plus standard of care in patients with mild COVID disease without comorbidities. Ang haba, no? Ang shortcut version nito, EVICT. EVICT. Efficacy of Vitex Nagundo for COVID-19 trial. Okay, so we said it was 2-stage. So the first stage is a dose-finding study, and the second stage is the placebo-controlled trial. Okay, let's talk about stage 1, no? When we say dose finding, we wanted to find out, since this never has been tried for COVID-19 before, what would be the best dose for COVID-19. So this is an open label dose finding study. And the trial sites that were welcoming to us was the Quezon Institute. No? They were very accommodating and really helped us out for stage 1. The Quezon Institute and our own PMP. You're in Camp Bagong Diwa, the National Capital Region Police Office. We also enrolled a lot of patients from Camp Bagong Diwa. Okay, so for the first stage, since it's a dose-finding study, we wanted to compare high-dose Lagundi, which is 1.2 grams 3 times a day, versus the normal dose Lagundi, which is 600 milligrams 3 times a day. So which one would be efficacious and of course safe, no? Not causing adverse effects. So these are the specific objectives, which also shows the outcome measures that we wanted to see, no? So comparing the 2 doses, we wanted to see which had a better mean clinical recovery time to resolution of symptoms, improvement of symptoms based on the global evaluation scale for physicians and patients. And we wanted to see the percentage of patients who would progress from mild to moderate or severe. Also, we wanted to see what the adverse effects were. So the 2 arms, as mentioned earlier, the high dose, the 1.2 grams 3 times a day, we enrolled 40 patients, and the 600 milligrams 3 times a day for 10 days, where we also enrolled the same number of patients. So this is just a representation of the flow of the study. So those with confirmed mild COVID, aged 19 to 55-year-old, without comorbidities, meaning no hypertension, diabetes, etc., we would get their informed consent, get a complete history and PE, get their O2 saturation rate, their mu score, and their other signs and symptoms. We did baseline laboratories of CBC, liver function test, kidney function test, as well as random blood sugar. And they would be randomized to either high dose or normal dose Lagundi. And we would follow them up every 3 days until day 20. So on the bottom are the outcome measures, which I have already talked about earlier. So definitely this has not been an easy clinical trial to perform, no? So let's look at some interim data. Around 295 patients were screened for eligibility, but only 75 were randomized in stage 1. No? So many were excluded because they did not meet the inclusion criteria. They were either asymptomatic, or they were older or younger than what was needed, or had other comorbidities, or were already on other antibiotics or other drugs. So among the 75 randomized, 39 were in the high dose group and 36 were in the usual dose group. And all patients were followed up and are undergoing analysis for the statistical analysis. And of course, we had good follow-up because they were all admitted in these quarantine centers. So for stage 1, the Lagundi tablet and syrup were very well tolerated for both doses. None of the patients in the group progressed to moderate or severe, and we are still doing some statistical analysis here. So just a few tables, but we're still doing more in-depth analysis for the results. So for the mean clinical recovery time and time to resolution of influenza-like symptoms, It was very similar for the high dose and the low dose group, around 6.5 days. And this was not statistically significant. For the physician-rated and patient-rated global evaluation scales by day 4, more than 90% in both groups had improvement, and more than 90% also had improvement based on ratings by the patient as well as the physician. In terms of adverse events, those that were elicited, and there was active eliciting of adverse events, there was diarrhea seen in a few patients, and pruritus was also seen. No nausea or vomiting was seen, and diarrhea and pruritus were already noted as adverse drug reactions in previous clinical trials. 5 patients stopped treatment because of these events, but the other patients continued on because of improvement of symptoms. So of course, this was not a perfect trial, no? For the problems encountered in stage 1, we noticed that the patient sought consult already several days into the illness, no? So there was a long lag time from when the patient started feeling something until they reached the quarantine center, no? So some of them are already asymptomatic by the time they get there, so we could not recruit them anymore. So other problems, many patients were on unnecessary antibiotics, no? As we know, for mild and moderate patients, according to the DOH guidelines, if they are really found to be positive for COVID-19, antibiotics are not necessary because this is a viral illness. And of course, enrollment is unpredictable. There are times when there are many patients to enroll, and there are times when there are no patients to enroll. No? So we had to find solutions for this. We informed the LGUs, and lately there's much better coordination, no? As they get better in contact tracing and handling these patients, no? Which ensured faster coordination and placements of the patients in the quarantine center. Also, we had to increase the number of trial sites so that recruitment would be faster. So that was stage 1. What is stage 2? Ito na yung buto-buto, no? Kung anong magandang dose sa stage 1, this is an adaptive trial. We will carry on the best dose from stage 1 to stage 2, no? And this time compare it to a placebo. So stage 2 is a randomized double-blind placebo-controlled trial. Double-blind meaning both the patient taking the drug plus the physician giving the drug doesn't know which treatment the patient is on, either on Lagundi or placebo, no? So that bias is removed. So again, using the tablet and syrup, Lagundi near prompt formulation, so plus standard of care compared to placebo plus standard of care in patients with mild COVID disease without comorbidities. So these are the objectives again. The outcome measures are actually very similar to stage 1, except that here we will now be taking serial swabs to check their RT-PCR to see if there is earlier clearance, no, of the virus from the patients. So what are the 2 arms? One arm would be Lagundi, whichever dose was the best, plus standard of care, and we will be enrolling 100 patients, versus the other arm is placebo plus standard of care. No? So to eliminate the placebo effect, no, there should always be a placebo so that they are unaware, no, of what treatment they are receiving. So we will also be enrolling 100 patients. No? Now for the first 100, so 50 for 50 per group, we will be doing serial RT-PCR testing for this patient. So here's the flow diagram now for these particular stage 2. So again, patients with mild COVID-19 disease, 19 to 55 years old without comorbidities. Of course, a written informed consent will be taken. Again, baseline history and PE, O2 saturation, the mu score, the severity of symptoms, and the baseline lab exams as we did in the first stage of CBC, BU and CREA, ALT, AST, and RBS. Then they will be randomized to either Lagundi or placebo, both sides given standard of care. Now, again, same outcome measures, time to clinical recovery, time to resolution of COVID symptoms, improvement based on patient scale and physician scale, and what is the percent which will progress to moderate or severe disease, plus taking serial RT-PCRs to see if they will clear the virus, in which side will clear the virus earlier. Of course, we want to look into safety and look at the adverse effects for both groups. So, stage 2 is presently ongoing. We have increased our trial sites, not just the Quezon Institute and the PNP NCRPO Special Facility Center in Taguig, but we have also been able to get permission to do our trial in the Valenzuela Quarantine Center, the Rizal High School, and the Silungan Molave in UP Diliman. And so far, 189 patients have been screened and 56 patients have been enrolled. Okay, just a little more pictures of our team going in and out of QI. No? So we also like to thank the administration of the trial sites that we were in. So just a few pictures of while we were doing stage 1. So here we are with some patients and taking down their data dito sa mga electronic CRF, and also teaching them how to take the drug and how to answer their diaries. So our expected date of completion will be in April this year, and of course this will all depend on our recruitment rate. But if it is going the way it is now, then looks like we are on track. So this is our team. We have a pharmacy team and a trial site team who goes there, and of course their safety is on the line every time they go there. No, so it includes Dr. Jaime Purificacion, Daisy Bagawisan, Ruth Elio, Dr. Shelly Heredia, Paulo Serrano, and supported by Bong Cervantes. And other parts of our team, no, Dr. Aina Castro does our data management and statistics. JM Velasco is going to help us in interpreting our RT-PCR results. And AJ Sobrevinas is our admin support. Our pharmacy team, Ma'am Joy Palakpak, Esel Talosa, and Jade Rodriguez.
So hello and again, good evening. Thank you, Dr. Marabel Sarte, for the interim results of the Lagoon trial. Now we proceed to the open forum to our participants. If you have any questions, again, please feel free to type in the question and answer box at the screens below. So far, we have a number of questions directed to Dr. Dairit. First is the from Ping Peria from General Santos City. Dr. David, he asks if there was a feature on GMA TV which interviewed a medical doctor who said that ingesting VCO frequently increases cholesterol. They want to confirm if the medical doctor is correct. And related to that, we have several questions.
Well, maybe to answer that question, the usual claim is that it raises your LDL, but actually it raises both HDL and LDL. So the ratio of LDL to HDL is retained. And now regarding raising cholesterol, none of the rise in cholesterol that was reported really makes it, it's not, I don't think it's harmful. Like if it goes up by a few points, it's usually those who don't understand, not are shall we say not very supportive of VCO, that they focus on increasing cholesterol. But I think the more important question is that does it really increase it to any dangerous level? And none, it really does not. So the cholesterol worry, I think, it's not it's not something to be worried about.
Thank you, Dr. David. Another question is related on VCO compared to MCT oils, which are commercially available. At the same time, another question from Mr. Carl Joseph Pablo was asking about the use of coconut oil instead for cooking. Between VCO and MCT oils and coconut oil, would these latter oils be as effective or viable for COVID, either as a food or treatment?
Well, in principle, they should. The MCT oil, you'd have to look at the composition, how much lauric acid there is there. That's on the assumption that it's only lauric acid that's effective. VCO does have other natural ingredients like phenols, although these they haven't really been tested, there, whether they have any efficacious effect against COVID. Now for your other question.
Okay, I think the last one was about the use of coconut oil for cooking as an
alternative. Well, I think it would be the same sort of answer. Actually, the first clinical trial of coconut oil against HIV, and it was before VCO came around. They used cooking oil. Now, I think between cooking oil and VCO, I think VCO would be much more pleasant to take. And anyway, no clinical trials using cooking oil have been done. So, the trials have been on VCO.
Related to the trials, how many, we are also being asked how many ml of VCO should be taken as a treatment, or at least as part of the treatment for COVID-19.
Yes, well, the clinical trial that was done, did a calculation of the number of ml per weight of the person. In general, it's about 3 to 4 tablespoons a day. It was distributed over 3 meals. So on the average, about 1 or so tablespoon after each meal. So take 1 tablespoon 3 times a day.
Okay, thank you, Dr. David.
Thank
we
you.
have another question, directed to all our speakers. What inspired them to pursue these medicinal plants and stick with investigating their properties for such a long time?
Maybe I'll ask Dr. Lazarte to answer first.
Okay. So actually, of course, everybody has been affected by this COVID, no? And for many of us, this has been very personal. And very early in this pandemic, we've lost loved ones, we've lost people. I lost my mentor to this disease. Everybody knows Dr. Sally Gatchalian. And, many of us were thinking, how can we help really in this fight against COVID? And one of our co-researchers, Dr. Vanji Amor, forwarded to me a article on the in silico testing of Lagundi, that might have a potential to be antiviral. And I researched it more. I found another article that possibly, it has a place, and looking into the other properties of Lagundi where it can help, no, in symptomatic treatment of this infection. It's really drove us not to pursue conducting a clinical trial, which is not easy. Paolo, it's not easy. You're out there every day. Even just, putting together the trial is really not easy, to get the funding and the support and all that. But really, the sheer desire, no, to really help in this pandemic is what has inspired us to do these studies.
Yeah, well, maybe I think, as I have mentioned in my presentation, even in last year, a year ago, and the end of January, I was already aware that VCO does have antiviral and antimicrobial properties, and why not check it out against COVID, and it gave some pretty promising results. And of course, coconut is a very important product for the Philippines, for coconut farmers, and so it's something that's quite affordable. And it's something that I've been taking VCO for about 20 years, so I'm not worried about my cholesterol or anything. So my cholesterol is actually fine, so no problem with it. So give it a try. Looked up the literature. It seemed to justify the use. So the clinical trials were, I think, justified, and it's been giving us good results.
Thank you. Any more questions from our...
How about where's Pip? You have to ask Pip also. Philip?
Yeah. So thank you very much, Ses. Thank you very much, Sir Toby. Thank you, Pam.
Thank you, Pip.
The reason mainly for Tawa-Tawa is the main reason, or the main motivation here is Urbanex Lab, which produces the capsules or the preparation for Tawa-Tawa, is actually based in Bacolod City, which is part of Region 6. So this is a way also to help the local R&D picture here, or environment for Western Visayas, so that other formulations or other companies that are marketing herbal formulations would also be given a chance, because there's also, as you mentioned, in literature, there are also promising studies that prove that Tawa-Tawa, it can be also antiviral. So for me, it's a very local. It's a local motivation to promote the R&D capability of Western Visayas. So that's it.
Thank you, Dr. Padilla. We have okay.
Go ahead, Paula. Thank you.
Sorry, we have an additional question from Alan Gran. So he asks, can we take VCO, Tawa-Tawa, and Lagundi in a day for the asymptomatic and symptomatic? And they're asking about the dosage and the potential use of combining the three.
Oh, why not? None of them are contraindicated against each other. So, for me, I wouldn't hesitate to take it if you so desire.
Well, Lagundi so far is not seen to have drug interactions with other drugs so far. I mean, based on the post-marketing reports from the FDA, since it's been in the market for how many years now? More than 20 years, I think. It has been seen to be very safe. Although with Tawa-Tawa, I don't know, because there have been no studies so far. So Yon. Pip, what do you think? Wala si Pip?
No, no.
Okay.
Actually, well, of course, I've been taking Lagundi whenever I have flu-like symptoms, and I take a lot of it, and it's actually a great it's a great
Of course, Sir
cure.
Toby, you discovered all the constituents, most of them,
No,
which are
but seriously, if I have like flu-like symptoms in the
yes.
morning, and I have a class to give in the evening, I take it in the morning, and I can just keep going the whole day, and it really works. It works, certainly works for me.
I'm afraid when I don't have Lagundi at home, I feel insecure because I also have allergic rhinitis. I use it all the time, and our team who goes to the trial centers have a free for all for the Lagundi. Take take Lagundi so that we hope it's protecting you also when we're out in the field.
Yeah.
So it's very safe, and we've used it a lot.
Okay, so for our participants, if there are no other questions, again, we'd like to thank our panelists for sharing with us their expertise, their interim results, and for the different studies. We would like to present to them our certificate or of thanks. Now, the Institute of Herbal Medicine, National Institutes of Health, University of the Philippines, Manila, presents this certificate of appreciation to our speakers, Dr. Philippine Padilla, Dr. Fabian Dairit, Dr. Cecilia Maraba Lazarte, in recognition of their invaluable contribution as speakers at the third Philippine Herbal Medicine Summit with the theme Ensuring Quality of Herbal Medicines Through Research, Regulation, and Cooperation, held on January 25 through 27, 2021. Again, we'd like to thank everyone for attending and for being present tonight, for sticking with us throughout all of the many lectures and sessions. Again, we'd like to request everyone to fill up the day one form. You can access it through the link seen, and we'll also be sending it through the chat box. You can click on the link for the evaluation form. Only those who have filled up the form will receive the certificate of attendance. Again, so also we would like to thank our organizers, our co-organizers, Plemex Vitex, Negundo by Trevenod, also to our sponsors, the Philippine Institute of Traditional and Alternative Healthcare, Binhi Biofarm, and New Market Link Pharmaceutical Corporation. We would also like to thank again UP Manila and the IMS UP Manila team for the support and assistance in holding this webinar. Again, thank you everyone. Good evening and see you tomorrow for the next session. Thank you.
Yeah, thank you.
Sir, stay on. Picture lang tayo.
Sige.
Can we ask the panelists to stay on? Pip, nawala ka. Nawala ka ba?
I'm here.
Andun siya.
Ayun. Okay. Ma'am Leslie, open your cameras. Is Prof here? He left na. Si Eric, nandito pa ba? Ma'am Leslie. Hi, Ma'am Vanji.
Yan si Vanji.
And the paper you gave me. Oy, Daisy May, asa na ba kayo? Lumabas kayo dyan. Oh,
Nandito lang, ma'am.
Ha?
Nandito lang po.
Nakapambahay ka, no? Game. One. Ay, si Ma'am Jessica Ray nandito. Ma'am, gusto niyong lumabas? 1, 2, 3, smile. Ay, yan dyan si Eileen at si Jade. Smile. Ayan, Ma'am Leslie. Hello. Thank you po, everyone. It was a good day.
Thank you.
A lot of them stayed on till the end.
Congratulations.
Thank you po. Thank you, Sir Toby, Pip.
Sige, thank you.
Congratulations. See you
Sige,
tomorrow.
bye-bye. Thank you.
Thank you. See you tomorrow, Ma'am Banj.
Good night.
Good night.
talaga bukas na lang.
Super good job.